Brian Kraemer. So Brian is an associate professor at the University of Washington. He's interested in protein aggregation in neurodegenerative diseases, has a particular interest in tauopathies and TDP-43. I think he's probably maybe best known for leveraging the awesome power of C. elegans. And hopefully we'll hear about his forward genetic screen for suppressors of TDP-43 toxicity. If not, I recommend reading his work. It's really absolutely fabulous. So Brian, it's wonderful to see you again, and it's all yours.
Thanks, Bob. I really appreciate the introduction and the opportunity to present some work from my group. In particular, I'd also like to thank the organizers and the previous speakers for filling in so much of the background. I think we can move through this quickly and maybe get to extra questions towards the end here. So today I'm going to tell you a story about one particular approach or screen that we've taken using C. elegans to try to get a handle on some of the neurodegenerative disease mechanisms and potentially novel targets for therapies that might translate. And so let's get into it. As you, I'm sure, are aware at this point, but I think it still bears saying, laboratory science these days is really a team sport. And this is my team. In particular, I'd like to highlight two women that have done the real heavy lifting on this screen, and that is Ali Saxton and Nikki Liachko. So,
hey,
yeah.
I'm sorry to interrupt, but we cannot see your slides yet.
Oh no. Well, let's see if we can fix that. Just a sec.
Okay.
That is good to know. Okay, so that's the team. We also, of course, appreciate our funding agencies and collaborators that have really made all this possible. So as you've heard this morning, TDP is one of the major disease proteins in ALS. Even in most familial forms of ALS, not all, but most of them, TDP-43 aggregates. So approximately 95% of cases are characterized by hyperphosphorylated abnormal TDP. And this is not merely a hallmark of motor neuron degeneration, but can rather be the cause, at least in a number of familial cases. Mutations in TDP drive the disease process. And as you've heard, of course, TDP-43, in addition to its pathological aggregation in disease, plays a number of essential roles in RNA processing. So loss of function of TDP is not a good thing as well. But in my group, the way we go about trying to study TDP is in a translational approach that, much like Daniela laid out for flies in her fly to human approach, we're taking a worm to human. And really in my lab, this starts in C. elegans as the primary discovery platform, and we've made some transgenic strains overexpressing human TDP that I'll talk about a bit more before we get into the screen. Things that come out of those screens that look interesting, we will try to translate a subset of those to human cellular models of TDP aggregation. And hypotheses that seem to hold in both worms and human cells, we will then take to preclinical mouse models of TDP-43 proteinopathy. And, so you may wonder, why start with C. elegans? And the simple answer to that question is for the experimental advantages, which are many. Probably the most important for aging-related neurodegenerative diseases is the rapid tempo of experimentation. You can have an animal mature from egg to adult in about 3 days. Normal worms live up to about 20 days in the lab, and TDP transgenic worms live less than half that, less than 10 days. So that allows a very rapid turnaround time in terms of studying aging-related phenotypes, which is very convenient. In addition to that, the C. elegans nervous system has been mapped with unprecedented accuracy at the synapse level. So about 300 neurons make about 5,000 synaptic connections, and that has been worked out at the it's essentially invariant, worked out for the entire adult nervous system. Cell lineage is well characterized. They have a simple and relatively easy to assess behavioral repertoire. Very importantly, they're optically transparent, so you can look inside of live animals routinely. And we have a lot of transgenic and genetic tools for manipulating the genome. Essentially, if you can think it, you can make it. And so that's really been convenient. And for today's talk, we're really going to dive into reverse genetics is the main discovery approach. So first, a little bit about the model that we made. So we've taken the human TDP43 gene and made its coding sequence be expressed in all C. elegans neurons, and shown over on the right here. You can see 3 different worm strains. When animals don't have any TDP, they move around plate normally, leaving these smooth sinusoidal tracks. If you express normal human TDP, they're not really significantly impaired. They move around pretty fine. But if you express TDP that has ALS causing mutations, this causes TDP43 to aggregate and be phosphorylated and ubiquitinated. And that causes a pretty severe motor phenotype. It's obvious on the plate. They don't move normally, they become paralyzed, and they die young. I don't really have time to go into all the details of the model, and it's been published for a while now. But the main features of the model are a series of behavioral or neurologic features characterized by early motor dysfunction that gets worse with age, loss or degeneration of axons, loss of motor neurons, and fatal paralysis that ultimately kills the animal. Now, they also exhibit accumulation of phospho-TDP species, which are probably the most consistent pathological hallmark of the disease in humans. And in this worm model, the amount of phospho-TDP correlates with the severity of the phenotype. So, of course, what we wanted to do was find out new things about disease mechanisms. And one approach that we took is a reverse genetic approach, where we wanted to ask what happens when we knock out gene function to TDP43 phenotypes. And to do that, we took advantage of experimental technique called RNAi, where in C. elegans, one can simply feed the worms in question different foods targeting all different genes in the genome. And so you make, their food is E. coli, and you make E. coli expressing a double-stranded RNA against the target gene of interest, and the worms that are fed that food will have reduced gene function. So this allows us to do experiments at a very large scale. And so what we did essentially was interrogate most of the genome, about almost 17,000 genes were screened, asking the question, does knocking this gene out make the RNA, does knocking this gene out make the TDP phenotype better or not? And what we really wanted to find was things that consistently made the TDP43 phenotype better. And from this screen, we identified 46 candidate suppressor genes that grouped into many different functional groups. About almost half of them had no significant alignment to human proteins. And so we don't really care so much about things that are distinct to worms, but wanted to look at things that are common between worms and humans. And the remaining classes were things like genes that are involved in energy production or metabolism, which you've heard a lot about already today. Genes involved in extracellular matrix or cellular structure and cytoskeleton. We also found genes involved in ion transport. We found a bunch of genes involved in nucleic acid function, like RNA binding proteins and DNA binding proteins and transcription factors. We found genes involved in controlling proteostasis, and we found genes involved in signaling. Now, we were particularly interested in signaling molecules because those are potentially the most druggable. There was also some genes identified that really are conserved between worms and humans, but their function is not really known, and so they're pretty hard to classify. We found a gene like that, I should say. So here's a list of all the genes. In red is the C. elegans name, and in blue is the proper human name for all of these genes. And so we're not going to really talk a lot about most of these going forward. I just want to put them all up there in case your favorite gene is here. We can talk about that in the Q&A or offline at any point. But you know, the issue with RNAi screens is you always want to really validate your findings with you know, authentic loss of function mutations that are, you know, much cleanly reducing gene functions. You want strong loss of function mutations. And for every gene in that list that we could find a strong loss of function mutation, we made TDP transgenic with that gene knocked out. So this is a motor function assay, just measuring motor velocity as a measure of radial dispersion. And what we saw was that many of these genes do show partial suppression, that is improved motor function in these TDP transgenic animals when we knocked out the target genes. Now that's all excellent, but of course we also wanted to see impacts on the accumulation of pathological TDP species. And in C. elegans, we know that phospho-TDP really is a toxic species. If you prevent phosphorylation, TDP is no longer toxic. And so things that are rescuing TDP phenotypes, we would expect to improve phospho-TDP accumulation. Shown here is some quantitative Western blotting for a variety of these candidate genes. So, you know, to summarize some of the more interesting results, we have a collection of genes that clearly positively influence motor function in the TDP transgenic animals, and also a few of them change phospho-TDP protein accumulation. And we're really going to focus in on this HSE5 molecule or gene because it is well conserved in humans and is involved in modulating signaling through the extracellular matrix. Now, you know, one of the problems with doing a big screen like this and getting a whole bunch of hits is you have a lot of work in front of you to characterize them all. And we have by no means, you know, even scratched the surface of all these genes, but I'm just going to follow up with one more interesting modifier that we'll spend the rest of our time here on. And so this gene is a gene called HSE5 in C. elegans, or GLCE, which is a modifier of heparan sulfate proteoglycans. And this is a key component of extracellular matrix and is involved in axon pathfinding. So let's talk a little bit more about that. So the human protein is GLCE, and that modifies heparan sulfate proteoglycans, which are moieties that are attached to cell surface proteins. And typically, HSPGs are involved in things like basement membrane extracellular matrix functions, so the formation of the stuff outside of cells that hold together tissue. HSPGs on these cell surface proteins frequently interact with growth factors or other signaling molecules to facilitate cell-to-cell contacts, cell to ECM interactions, things like cell migration, anchoring of tissue, these sorts of things. So they're very important for participation in cell signaling. So GLCE is an enzyme that conducts heparan sulfate C5 epimerase reactions. So this is essentially inversion stereochemistry where a carboxylic acid and a hydrogen are flipped positions on sugar moieties. So this is a glucuronic acid to a glucuronic acid epimerization. The end result of this is an increased flexibility of the heparan sulfate moieties, which can promote more signaling activity. Now, interestingly, mutations in any of the three C. elegans heparan sulfate modifying genes or modifying enzymes are encoded by genes HST2, HST6, and HSE5. And mutations, loss of function mutations in any of those three genes cause axon pathfinding defects. In particular, HSE5 disrupts motor neuron axon pathfinding, so you get incorrect pathfinding phenotypes. We'll talk about that a little more, but so far I've shown that HSE5 can promote rescue of motor phenotypes and decreases the accumulation of phospho-TDP. But probably one of the phenotypes we're most interested in the context of ALS is there a way to extend lifespan? And so unfortunately, when we looked in these HSE5 loss of function worms, that does not rescue the lifespan of TDP43 phenotypes. And we're not exactly sure why that is, but that is an observation that we've made. Another thing that we've observed, which was very surprising to me, is that HSE5 loss of function does not protect against TDP43 driven neurodegeneration or neuronal loss. So it's rescuing the motor phenotype without rescuing neuron loss. And that really confused us for a while. But when we look more closely at neuronal connectivity, one of the phenotypes in a regular worm that's different in the HSE5 mutants in the absence of TDP is that HSE5 causes branching and essentially extra connectivity of neurons in the HSE5 mutant. And what we see in these TDP worms, despite the fact they're undergoing neurodegenerative changes and losing neurons, they also are showing extra connectivity of their axons. So it would seem that the additional branching and additional connectivity is helping restore motor function in the absence of some or dropout of some neurons. So that was a pretty surprising finding. And of course, one of the things you care about an awful lot in these sort of studies is does it translate to the human condition? And while we're not going to get all the way there today, we did at least start to go down that road and look at what happens in human cells. So if we take human cells, we can trigger the accumulation of phospho-TDP by treatment with a small molecule called ethacrynic acid, which depletes glutathione and causes oxidative stress. And so under EA treatment, we get a nice abundant phospho-TDP accumulation. And if we knock down the human GLCE, we see a reduction in that phospho-TDP accumulation. This is very robust and reproducible. So, human cells in a dish are an awful long ways from a human patient, but this is at least supporting the idea that there is conserved function in phospho-TDP pathways. Now, this doesn't provide us any insight into the mechanism by that protection, and I'm not going to, unfortunately, get to that today because we just don't understand it yet. But that is a thing that we have hope for going forward, is that we can find ways to translationally address this target, maybe with small molecules. So we also wanted to look in the human disease state, and so we took a look at human TDP43 proteinopathy, and this is frontal temporal dementia with TDP inclusions. And what we see is that in normal patients that have been studied at autopsy, there's abundant GLCE expression in the brain. And if we look at GLCE in FTLD patients that have TDP43 proteinopathy, despite the fact that they have abundant TDP accumulations, they have lost much of their GLCE. And this is a statistically significant accumulation that we've seen in like 15 or so FTLD-TDP cases. So we think that there is some action there with GLCE in human TDP43 proteinopathy, and that perhaps this loss of GLCE is a response to the accumulation of pathological lesions. But again, we don't really understand the mechanism of this yet, but it does provide evidence of translation. Okay, so I'd just like to summarize what we've seen, and then we'll take some extra time for questions. So we conducted a genome-wide screen and uncovered many new suppressors of motor dysfunction and phospho-TDP accumulation in C. elegans. One of those is translated, and that is HSE5. When we knock out HSE5, we see rescue of TDP motor dysfunction in C. elegans, and we see decreases in phospho-TDP accumulation. We've also shown that knockdown of GLCE decreases phospho-TDP accumulation in human cells, and that GLCE becomes depleted in human TDP-43 proteinopathy disorders. I'd like to thank you for listening, and I'm happy to take your questions.
Great. Thank you, Brian. Okay, so turn off your share screen just for a sec.
Okay, let's see. Stop share. There we go.
Okay, cool. Let me ask you this. So, are there drugs that can target the epimerase?
Not really.
You would think that it's enzymatic. You think it's maybe targetable?
I would hope so. You know, we've started to work on developing small molecules targeting RNA binding proteins, which is, you know, supposed to be harder than targeting enzymes, but we haven't really got the assay set up yet. You know, I think ideally I'd like to see some mouse data before we go, you know, whole hog on trying to do small molecule discovery. But I do think that's the logical step once you have, you know, some more convincing mechanistic and translational data, which is what we're trying to do right now.
And how many GLCEs are there in the mouse genome?
I believe there's one.
There's one. So, and does the knockout live?
I don't know that that's no. I don't think so. And you know, I think ideally you'd want a conditional, right? I don't think that is going to be compatible with development, for example. You know, just based on what we know about HSPGs and other developmental pathways, you know, cell migration generally gets all messed up when you do that sort of thing. So I think that's probably something that you need to do conditionally, and that experiment just hasn't been done.
Well, maybe something like antisense oligos would be.
Yeah, that you know might be another way. You know, at least in the worm model, though, we really need to see a pretty robust loss of function to get phenotypes. And so, you know, with you know, antisense, you really have, you know, that's usually a partial effect, right? And so, I'm not sure that that's going to be sufficient loss of function to really rescue. It's hard to know though. You know, we're just really scratching the surface on all of this stuff, and that is, you know, the downside of these big screens is you find yourself in a new place that nobody's really ever been before, which is great, but then there's so much work to do, you know. And so, you know, in a screen like this, it's really kind of an effort to recruit others to you know, to start studying it, you know. So we just put it out there and see what happens. And, you know, when we did this sort of thing for tau, it was amazing the number of different labs that contacted me. Oh, we've been working on this gene for a couple of years now, and this is really important. And, you know, you just don't know until you put the story out there. And so this is published fairly recently, so we don't know much yet about who else is on the same page with us.
Which gets to another question I had, and it's also in that question and answer box, so I can speak for both of us. Have you looked at the effect of loss of function of the epimerase on other models of neurodegenerative disease?
We did a little bit with tau, which is probably the other neurodegenerative model, and we didn't really see much. But you know, we haven't done an extensive survey. You know, I think it'd be interesting to look at polyglutamine, for instance. But you know, it does seem like it might be relatively TDP specific. And most of the modifiers we find tend to be that way, although sometimes, you know, if you're into chaperones or, you know, other general regulators of proteostasis, you'll frequently find that they're all modified. So, but it's always an intriguing question, you know, what gives, you know, cell type specificity or, you know, selective vulnerability.
Right. There was another question in the Q&A that says, which classes of motor neurons are mostly affected in C. elegans and the HSC loss of function mutants? Does that tell you anything about the underlying biology of TDP-43 by studying these neurons specifically?
You know, our favorite class of neurons to study in C. elegans is the GABAergic inhibitory motor neurons. And I think that those are a disease relevant population to study for ALS. But, you know, it's mostly a matter of convenience that we look at them. I think that a lot of the reporters, you know, the GFP reporter lines, while those neurons are intact in wild type animals, as soon as you add an additional insult like TDP or loss of function of another mutation, you run into viability issues. So, you know, we haven't really studied the cholinergic motor neurons because you essentially kill the worm if you put that reporter in the background, at least so far. You know, I'd like to find very specific, you know, like maybe egg-laying motor neuron reporters or something like that, where you can lose them and still have a live animal. But we focus on the GABAergic motor neurons because they're convenient and they're really actually dispensable for viability. So it's a more convenient population to study.
You have an UNC phenotype that you can score.
Yeah. Exactly. Yeah, very visible plate phenotype.
Cool.